I'm going to record. Okay, so we are going to now do that review that I mentioned, and I am going to just see if I can control this. Okay, good. So here's the review. Remember, it's just the same thing that we talked about before. This is not a test. I don't like it when anyone thinks it's a test. That's not the point of any of this. It's just to get you to understand things better. And it's the same stuff we've seen literally like slide for slide, but we're just like putting them out of order. So you just kind of can tell kind of if you know something or not, okay? It's good to not forget this stuff. Okay, so here's a name the layer, which I think is like the whole reason why we do this, right? So here's a name the layer. I'm just gonna point to it. You tell me what it is, okay? What is that layer, if you remember?
It's the inner plexiform.
Easy, right? So the inner plexiform layer, okay? Tell me what the non-neuronal cells are in the retina. You remember I introduced 7 of them?
Yeah.
2 of them are not neuronal. Which ones are they?
So the Mueller cells are not neuronal.
Yep. And what's the other one? I made them both green always. I don't know if you remember. So yes, so you're correct. Start naming retinal cells. Just no, I mean, that's how you do it, right?
The amacrine cells.
Right, that's a neuron.
Horizontal cells.
That's a neuron.
Ganglion cells.
That's a neuron.
Bipolar cells.
That's a neuron. You're getting them almost all. And then the photoreceptor. I'll give you the last one. And then what does a photoreceptor stand on?
The RPE.
You got it. The RPE is not a neuron. It's an epithelial cell.
Yes.
Right? So you got it. So those are your 2. Perfect. All right. Tell me what the main neuronal pathway is, right? Like what is the classic photon comes into the retina and then it gets to the brain? Like how does it do that? What is in the retina? What are the cells that are involved in that main chain?
Okay. So the light comes in, they get absorbed by the photoreceptors.
Yep.
Then the photoreceptors transmit signals or don't transmit signals, but then that goes to the bipolar cells.
You got it.
Then to the ganglion cells.
You got it. And there it is, right? Photoreceptor, bipolar, ganglion cell.
Yes.
Perfect. Here, we're going to name a layer again. And then you're going to tell me like why it looks the way it does. Okay. This is always like, this is a harder one, but you remember this particular kind of image, right? So we're talking about this layer, the thin one that we tend to peel. What is that?
The internal limiting membrane.
Correct. And what is it made up of?
It's made out of like glial tissue.
You're not wrong, kind of, yes. But what is specifically what? Well, I'm sorry, I should be specific. Like, why does it look the way it does? What cell makes it up? Right? It's not a neuron. I'm giving you a hint.
Is it like the Mueller cell?
Yeah. And what do we call is it the head or the feet?
The feet. Good.
The foot plates of the Mueller cells. You got it, right? Internal limiting membrane, light. Okay. Name the layer. What is that dark layer?
That is the outer neuronal layer, outer nuclear layer.
You got it. Outer nuclear layer. Perfect. Remember I called them the summation neurons. They're like the helper neurons. I make them red. Okay. They're not the main chain neurons, right? What are those? They're neurons, but they're not the main chain.
The amacrine and the horizontal
cells. You got it, right? Horizontal and amacrine. I make them red just so that it makes sense in your head. Okay. Completely different part of the situation, right? We're in the primary visual cortex. I think you remember sort of what we're looking at. I'm pointing to those 2 layers, okay? What is the pathway that you'd think would terminate in that layer? Do you remember?
I think that was the coniocellular pathway.
You killed it. Yeah, exactly. The coniocellular pathway, right? It's the add-on pack. Do you remember these guys, the cytochrome oxidase blobs? You remember that? Okay, so you got it. And it's the bistratified ganglion cell coniocellular pathway. Perfect. I don't know if I did this one with you. Maybe I did. No, I mean, I'm just saying, what is the full name of this scan? What is this scan that I'm showing you right now?
Is that, I mean, those are A scan.
A scans. Good. You got it. Now, what does A stand for? Do you remember? Did I go through this with you?
Was it, I think they're like aggregates or something?
Well, aggregate is one word that starts with an A. It's amplitude, right? Do you remember amplitude?
Yes.
Do you remember? Yeah. So A stands for amplitude, right? And so remember, it's because you're reflecting off of different acoustic properties, right? That's why it's a sound wave going through. And anytime there's a different acoustic property from one spot to another, you get a bump here, you get a spike, right? So that's what's being returned here. That's the amplitude that's being returned. So we called it the amplitude scan, just so you know, right? An A scan is not named A because it's the first one, although it works out that way, okay? Amplitude scan, back to name the layer, okay? What is that layer?
The nerve fiber layer.
Good, nerve fiber layer, cool. Here's another one. What is the full name of this scan? What do we call that?
That's a B scan.
And what does B stand for? Do you remember?
I don't remember.
It's called brightness.
Brightness.
Remember?
Okay.
And why is it brightness? Because it's a bunch of A scans and then you tip them over 90 degrees. Do you remember me showing you that?
Yeah.
You tip them over 90 degrees and then where the spikes touch like the screen, it shows up as bright. And so that's why they call that the brightness scan. It's not important to maybe remember exactly how it works, but remember brightness scan, that's what B is. Amplitude scan is for the A, and you take a bunch of A scans, tip them over, and you get the B scan, the brightness scan in 2D.
Yeah.
How many different cones and rods exist in the human retina? How many different rods are there? Do you remember? Or how many rods? I shouldn't say different. That's the incorrect word. How many rods?
There's like 120 million total of the both combined.
No. So you actually got it correct, except yes. So 120 million rods, 6 million cones.
Yeah.
So 126 million total. Make sense?
Yes.
Cool, but you were right. It's in the 100 millions, right? So you got it. It's basically 10 to the 8th, right? I mean, that's the idea. Here we go. I am back in the brain again. Because we're going whiplashing all over the place, right? What is that layer, first of all? What is that layer called? Just say it for me. It's layer what? I'm pointing to it. It's right there. What is it?
4C beta.
4C beta. So what is the pathway that terminates there?
I think that was the parvocellular.
Parvocellular pathway. Tell me exactly, what is the ganglion cell called that's associated with that?
The oh my gosh.
You're there. You can feel it. Remember, it's the opposite letters, right? M goes with P and P goes with M. So you've got a P here. So it's parvocellular. So it's got to be an M type ganglion cell. What does the M stand for? You got it. Midget ganglion cell. And what does it deal with?
So that's like blue light. Oh, no. I'm wrong. Sorry. That's like red green.
You got it. Red green. Exactly. What is blue though? Who does blue?
That's coniocellular.
You got it. Coniocellular, bistratified. Exactly. Right. Cool. Name the layer. Back whiplash. Back into the retina. What is that layer?
That is the inner plexiform.
Try again.
No, sorry, the ganglion cell.
Yeah, you got it, right? I mean, it makes sense. I even thought that half a second when I looked at it. And then I'm like, oh, no, it's a little bit closer. Ganglion cell layer. Perfect. Here we are in the schema here. This is for a photoreceptor. In the dark, is it active or inactive?
It's active.
Active. Tell me what's its neurotransmitter that it's secreting.
It's releasing glutamate.
It's releasing glutamate. And then, of course, this is inhibited. So glutamate, no glutamate. Perfect. Here we go. Back to the retina. Name the layer and what's in it. This layer right here, this dark layer right here.
That's the myoid.
Everyone does that.
No, it's not. It's the...
What is this? You got it. You're close.
Oh my gosh.
Okay, so let's walk ourselves through it, right? This is a photoreceptor.
Outer photoreceptors.
Yes, well, the outer segments. You got it. Outer segments of the photoreceptors. Don't ask me why that's this weirdly beige. I don't know why I did that, but outer segments of why are they dark? Because they accept the photon, right? And when you pull a photon in, you're not reflecting it. And so if you pull it in, you're going to get a black hole effect. It's going to be dark. If you reflect, it's light. And then on top of it, the cones and the rods, they go away from each other. They kind of fade away. So you're not getting reflectivity in that layer. Make sense?
Yes.
Okay, good. Another way of looking at the primary visual cortex, okay? I'm going to point to something. I'm pointing here, okay? What layer is that, first of all? Is it alpha or beta?
It's probably beta.
You got it. It's beta. Thus, what is the pathway that corresponds to that location?
Parvocellular.
Parvocellular pathway We talked about it, midget ganglion cell, red, green. Perfect. Okay. This one's harder, but approximately how many bipolar cells, if you were to add all of them up in total, exist? There were 126 million photoreceptors. Tell me,
36
you
million?
got it. 30. What'd you say? What did you say? Sorry, Jeanette, say that again.
36.
You got it. 36 million. Yeah,
Yeah.
exactly. Right. So it's, I only putting this out there because you should know magnitude and then you should know the pyramid structure, right? So it's going 1 to 4, basically, right? There's one bipolar cell to four photoreceptors. Name the layer. What is this white layer right here?
That is the outer plexiform.
Yep, outer plexiform. Can I just point out something? Notice how the actual retina right now is not the hard part. Right? When you started all these lectures, I'm sure you're like, I don't even know if I'm going to know the layers. And now you're just kind of like, yeah, I can get through that. I'm kind of like answering all these other questions right now, right? So it's pretty cool. Here's a primary visual cortex again, okay? I'm just randomly putting it way up here. If you had to guess which pathway would terminate there, okay? It's not a canonical spot of the brain, but what pathway would you guess? What is near to it? This is 2 and 3. You told me this was which one? What pathway?
That's probably coniocellular.
Correct. So I would guess the same thing, probably coniocellular, and you got it right, bistratified. And you see this empirically. Okay, remember this now. Okay, now we're here. This is your LGN. Okay, I'm going to point there. You tell me what pathway, what ganglion cell, and where is it ipsilateral, contralateral, and what it carries. Tell me right now.
Okay, so it's layer 5. So that's going to be an ipsilateral, parvocellular, so midget ganglion cell.
Which means what in terms of functionality?
Which means that it's involved with red-green color.
Perfect. Exactly. Approximately how many ganglion cells exist? Total.
1 to 2 million.
You got it. 1 million. I say 0.7 to 1.5, but you're exact. Pick 1 million, you win the game. Okay. So it's 1 million ganglion cells, 36 or 30 million, you can say 36 million bipolar cells, 126 million photoreceptors. That's the pyramid. Okay.
Yes.
Name the layer and what's in it. I'm pointing to this layer. Okay. What is that layer? It's easy, but what's that layer? What do we usually call this?
That's the Brooks RPE complex.
Correct. You got it. So, and why is it light again? Tell me.
Well, it's because there's like
so it's RPE.
Why it's light.
What are they joined by? Tight junctions, you remember? And so when you're tightly packed, you reflect with light, right?
Yes.
That's canonical kind of thing for OCT 101, right? If you're tightly packed, you reflect light. Okay. Now, why is it important that I bring this one up? Because what is the special nature of this one? There are 2 blood retina barriers. This one is the inner or outer blood retina barrier.
That's the outer one.
Correct. What is the inner one?
The retinal vessel walls.
Correct. Or the endothelial cell tight junctions around the vessel that make up the vessel. Exactly. So that's why I always ask the question for this one, because there's 2 sources of vascular supply to the retina. The first is the retinal vasculature. So you need a blood retinal barrier for that. That's the endothelial cells. But the second is the choroid, right? The diffusion of nutrients through the choroid. You can't just have it willy-nilly connecting to the, going into the brain. So what do? We don't actually have the endothelial cells around the choroid that are our blood retina barrier. We have the RPE tight junctions. Make sense?
Yeah.
Cool. Name the types of the ganglion cells. There are 5. Okay. Give me the 5.
So there's the parvocellular or midget ganglion cells. And then there are the coniocellular bistratified ganglion cells.
Good.
And then there are the magnocellular or parasol type ganglion cells.
Perfect.
Then the ones that are intrinsically photosensitive.
Retinal ganglion cells, you got it. And tell me, what is the thing they carry that makes them intrinsically photosensitive?
They have their own pigment.
They have their own opsin, exactly. And what do we call that? Do you remember?
The melanopsin.
You got it. Melanopsin. That's the opsin they carry. That's what its name is. You got it. Next one. What's the last one? It's like kind of a
It's like it's like the other.
other, right? Other, and it deals with saccades, right? You got it. Perfect. Exactly. Right. Exactly what you'd have said. Okay. Back here, I already did this one. This is coneus. I don't know why I should, but you know that you knew that, right? I mean, it's the add-on pack. It's on top. Perfect. And then there's our delineation. Back to the lateral geniculate nucleus.
Okay.
I'm pointing there.
So that is going to be a ipsilateral parvocellular or midget type ganglion cell that's carrying information about red-green light.
Perfect. Exactly. Okay. This is a horizontal cell. Okay.
Okay.
Here we go. So in the dark, is the central photoreceptor active or inhibited?
It's activated.
You got it. It's activated. Therefore, is the horizontal cell that's at that central photoreceptor's party listening to the same music and liking the same music? Is he active or inactive?
He's activated.
He's activated. Okay, good. And then, okay, so then it's inhibited here. So then in that top line, what happens to the surrounding photoreceptors in that top line? Are they active or inactive?
They're being inhibited by the
Inhibited. You got it. And then the opposite's true here. Perfect. Perfect. Back here, we're at this one.
So 6 is, so that's also going to be a parvocellular pathway ganglion cell.
You got it.
Which is contralateral this time, carrying
And then
information about red-green.
Red-green, midget, perfect. You got it. We're hammering this one because it's 80% of all ganglion cells. You know this, you should be able to figure out what type of cell I'm looking at right now.
Right.
Okay? I mean, it's like, you know, a different screen here and it's all flipped over, but tell me this particular cell, if you can kind of figure it out, whether it's off, if it's on, and then whether it's, and first of all, tell me what it is. What do you think this is?
Was, did D stand for diffuse?
Yep, diffuse, and then what? What kind of cell is it?
Beta.
Well, it's not a beta cell.
Or
What cells are we looking
bipolar.
at? You got it. A diffuse bipolar cell. So if it's a diffuse bipolar cell, okay, that means it connects. What is the diffuse referring to? Its connection to photoreceptors or ganglion cells? Which one is it talking about?
To photoreceptors.
You got it. Photoreceptors. So is it many or single?
Many. So
Many. Exactly. And is this on or off?
Off.
You got it. That's it, right? Diffuse off bipolar cell, many, normally cones, but it could be rods. Perfect. Now we're going to do this schema here. So it's, you know, you remember all this stuff and okay, I don't even know why I did that. Okay, never mind. We're just going to show this for no particular reason and move on. Okay, so we're going to go to the lateral geniculate nucleus again. I'm pointing at that one. Okay, what ganglion cell deals with that particular pathway? What is that pathway dealing with? Edinger-Westphal nucleus. That's called the pre-olivary.
The intrinsically
photosensitive. And why do you know that? What is this one dealing with?
Your pupillary light reflex.
Perfect. Pupillary light reflex. IPRGC, melanopsin, pupils in ambient light, or what have you. Perfect. Name the layer. What is that?
Vitreous.
King or queen, vitreous. Exactly. That's how you normalize, right? Back to this one. Okay, so I'm going to turn this off. Now you tell me, in the dark, is the photoreceptor active or inactive?
Active.
Okay, if that's the case, is the bipolar on or bipolar off active?
The bipolar off is active.
Correct. And then in that case, is the ganglion off or ganglion on active?
Off.
Perfect. That's your line. Okay, back to the primary visual cortex. Back to this one. Tell me what that layer is.
4C beta.
You got it, which is, again, just really fast. What is the pathway that terminates there? Parvocellular pathway. You got it. Red, green, everything. You understood. Name the layer. What's that one?
That is the internuclear layer.
Internuclear layer. Perfect. Here's another name the layer. Okay, we're going to point to this line, and I want to know why it looks the way it does. So what is this line?
It's the external limiting membrane.
External neural limiting membrane. What are the two cells involved with it? Do you remember?
I think they're also the Mueller cells.
Yes, Mueller cell is one, and then it's the
photoreceptors.
Photoreceptor, and it's a tight junction between them. Remember? You got it. So as I always say, the head of the Mueller cells here, the foot is over here, the feet connect together and the head connects to a photoreceptor. That's the tight junctions between the two, I suppose. Okay, so you have it perfectly. Here we go. We're gonna now do the light way. In the light, just belaboring the point, because now it's becoming easy, but it was never easy to begin with, right? But let's just belabor the point. In the light, is the photoreceptor active or inactive?
It is inactive.
Inactive. Then in that state, is the bipolar on or bipolar off active?
Bipolar on is active.
Correct. And then in that state, is ganglion cell on or off? Which one of those is active?
Ganglion on.
Correct. There you go. Perfect. Back here. This one is 4A. I don't know if I really want to test this, but I'll just say this to you. You remember these two would be talking. You remember I was trying to give you that idea that as you got up close to this, the colors needed a way to talk to each other, right? Yeah. That's why I'm just trying to give you that integration sense that's happening here. Okay, lateral geniculate nucleus. I am talking about this layer, which is not one.
Probably one of the coniocellular.
Okay, good. Coniocellular. And so where? From where?
From
ipsi or contra?
Contralateral.
Contralateral, right? Because 1, 4, 6, right? So contralateral, you said coniocellular, which is what type of ganglion cell?
Stratified.
Bistratified. And what does it deal with?
About blue light.
Blue light, and also like proprioception, right? And like sensory processing is like an extra add-on. Okay, perfect. Contralateral color blue integration. We're adding our favorite cells here, and we're going to do this now again. We're going in the dark. Okay. In the dark, is the photoreceptor active or inactive? The central one.
Active.
It is active, which means bipolar on or bipolar off is active.
Bipolar off is active.
Bipolar off is active, which means ganglion on or ganglion off. Which one's active?
Ganglion off is active.
Ganglion off is active. Now we're going to talk about the horizontal cell. In this state in the dark, is the horizontal cell active or inactive?
I think it's active.
It's active. So the surrounding photoreceptors are active or inactive?
Inhibited.
Inhibited. That's the line right there. Perfect. Okay. Now we're going to go here, and I'm pointing to that layer.
Okay. So that is the first layer. So that's going to be a contralateral. Information from a magnocellular paracell pathway ganglion cell.
And what does that deal with?
Oh my gosh.
Magnocellular
motion.
Motion, right? Motion, right? Right? So you got it. Okay. So contralateral, low contrast, but motion, right? We don't care about colors, right? We want speed, right? Okay. Yeah. We're going back to this one. Light. In the light, is the central photoreceptor active or inactive?
It's inactive.
It's inactive.
Inactive.
Correct. So bipolar on or bipolar off is active here?
Bipolar on is active.
Correct. And then ganglion on or off? Which one's active?
On is active.
Correct. So the horizontal cell, that's the same party as the central photoreceptor, is that active or inactive?
Inactive. Inactive.
And the surrounding photoreceptors, are they active or inactive?
They're going to be less inhibited, so they're active.
They're active. Exactly. There you go. It's just going to look like this. Exactly. Perfect. All right. What is this cell? Tell me where he starts, where he ends, and where his nucleus is.
That's the Mueller cell, and the foot plates are at the internal limiting membrane, and the head is the external limiting membrane. And its cell body is in the inner nucleus.
Yeah, its nucleus is exactly. You got it. Perfect. Mueller cell. Name the layer and what's in it. This is the light layer right here. What is it?
The ellipsoid.
Correct. And why is it called that?
Because it's filled with the mitochondria.
Yep, which look like ellipses. Exactly. Perfect. What layer of the retina am I talking about right now?
Wait, okay, so that's a
that's a photoreceptor. That's a cone.
That's a photoreceptor that is connected to a bipolar cell.
And then, so where is the bipolar cell terminating? Where do they normally terminate classically?
In the inner
plexiform layer. You got it. And then, just for extra credit, it's either on or off. Sublamina A, I'm just going to say, I won't say extra credit, is off, and this is on. It's just the distal one is on. But yes, this is the inner plexiform layer. right? It's just like, looking at it from a different angle, but you still know what you're doing, right? Nothing, I always say for these things, like nothing actually changes. It's all sleight of hand. We're trying to like color it differently. And I'm trying to do it so you understand it. But then in the flip, nothing has changed. The underlying content is always the same. Oh, you said it was a bipolar cell. Exactly. So you got that. And is it on or off? Right. So we said it was on because it's in sublamina B. It's further away. Okay. What is that cell? Hint, hint.
That's a bipolar cell.
You got it. And where does it start and where does it finish? And where is its cell body or nucleus?
Starts in the outer plexiform layer and ends in the inner plexiform layer. And the cell body is in the inner nuclear layer.
Perfect. Inner nuclear layer, bipolar cell. Who is this?
That's the photoreceptor.
Start and finish, and where's the cell body?
So it starts in the, I guess.
So this is the one that's the part that's going to be questionable, right? It's this. What's above it though? What's the layer? We actually named this layer, right? Remember the fimbriae of the apical.
The interdigitation zone.
Exactly, interdigitation zone. Where does it end?
In the outer plexiform.
Outer plexiform, and where's its cell body?
The outer nuclear layer.
Exactly, you got it. Okay, photoreceptor. Name the cell. Okay, yeah, okay. Okay, I'm like doing this with you. Okay, it's synapses with only one cone.
Midget,
so what is midget bipolar cell? You got it. It's synapses with multiple cones.
Was that the flat one?
Well, remember, if it's synapses with multiple cones, remember, what is the distinction? It's midget, or what's the other one? It starts with the D. Diffuse. Exactly. Diffuse, right? Diffuse. And then I slipped this in. Look, this statement is like, well, horizontal cell also, qualifies, right? I just threw that in there just to, think about it from a different angle. Like, who synapses with multiple cones? Oh, a diffuse bipolar cell does. We know that because it's diffuse. Yeah. But then we also know horizontal cells do. And we're not even talking about bipolar cells, just to kind of get you thinking. Okay, what you were talking about, bipolar cell into the synaptic ribbon, not on the surface. What is it when it's into the synaptic ribbon?
Oh, it's like invaginating.
Invaginating. And then what about on the surface, like you said?
The flat ones.
Correct. That's the dichotomy, right? It's flat versus invaginating, midget versus diffuse, right? So you can be a flat midget bipolar cell, and presumably you could be a diffuse invaginating bipolar cell, but we don't call it that. We actually call that the rod bipolar cell. I'll just put that out there. Okay. But like, that's the only one that I know of that's like that. But just know that this is how you would look at it, right? Okay, good. Mm-hmm. So LGN again, we're in that white layer.
That's the coniocellular.
And what is where? From where?
So it's in front of layer 2, so it's going to be ipsilateral.
Ipsilateral eye, correct? So it's in the same side of where this LGN is. That's where this ganglion cell would be. And what does the ganglion cell look like again when I go to that particular layer?
Bistratified.
Bistratified, carrying blue, perception, blah, blah, blah. Okay, good, perfect. What is that cell?
RPE Brooks.
Yeah, it's really RPE Brooks complex, RPE. You got it. Primary visual cortex. We're in what is that layer again?
So it's 4C alpha.
So who terminates there?
The magnocellular parasol ganglion cells.
Ganglion cell. And what does it carry? What information?
Motion.
Motion. Exactly. Perfect. Right over here. Tell me what that cell is, where it starts, where it finishes, where its nucleus is.
That's the amacrine cell.
Yep.
Starts in the outer plexiform and ends in the inner plexiform.
So back up here. Does it ever go in the outer plexiform? So basically what it does, inner plexiform is correct. But that's the only place it sits. I haven't gone through. Yeah.
With the photoreceptor. Right.
Correct. So it'll synapse with the ganglion cells, right? Because that's what an amacrine cell does. We haven't gone through it. Okay. So it's inner plexiform. You're correct. Where's its cell body?
In the inner nuclear layer.
Correct. So I just will say that's all it is, right? Because it's like a U. It connects from one ganglion cell to another. It doesn't like span, but it like connects. It kind of like, like a spider with its feet on like the ceiling, if you will, right? So it's constantly starts on the ceiling, ends on the ceiling kind of thing, right? Yeah. That's the amacrine cell. Name the cell labeled number 1. What is this? What is this cell? I don't know why okay. It only has a on bipolar cell. It doesn't have an off. It doesn't even connect to a ganglion cell from its bipolar cell. So what is this cell with a 1?
Is it a rod?
It's a rod. It's a rod. It looks like a rod, right? And and I said 1. And then there you go. And then there's amacrine 2. And you remember this whole thing, right? Where it hijacks the next door cone machinery's on cell, so that way it can transmit it using help from an amacrine 2 and that's what's called A2 cell. That's what the amacrine cell is called. We get into this later, but why is this important? Because I was trying to give you the idea of how to think about this clinically. If I axe first this rod bipolar cell, there's only one type of bipolar cell that interfaces with rods, as we know. So if I do that, my rods don't work, which means I have nyctalopia. My my magnocellular pathway doesn't work, right? Because it's gone. The guys who relay that information don't get to the LGN because this guy's dead. Or here's the other thing. If I kill genetically A2, the same problem happens. Make sense?
Yeah.
So it's a very it has a lot of failure paths, so to speak, right?
Yeah.
And so rod, and then I was saying this is the rod bipolar cell on top of it. I don't know why I wrote that. Okay. Who is this? Where does it start? And where does it finish in the retina? And then where is its cell body or its nucleus?
The ganglion cell.
Yep.
Starts in the inner plexiform. Terminates in the LGN. And
you're correct. It terminates in the LGN. Yep. But what is this layer that it sort of?
Nerve fiber layer.
Nerve fiber layer. And then what's the nucleus? Where is it?
The ganglion cell layer.
You got it. Perfect. Ganglion cell. Who is that?
That's the horizontal cell.
Okay. So it starts and finishes in what layer?
In the outer plexiform.
Correct. And then where is its cell body?
Inner nuclear layer.
Exactly. Perfect. Okay. Okay. So I'm telling you that this is the inner plexiform layer. So let's orient ourselves, right? I have divided this. Here is a line here in between. We know that was sublamina A and sublamina B. We also remember that this is closer to kind of where the bipolar cells are, right? Where the photoreceptors are, the inner nuclear layer. So we know that this is the off and the on. You remember on is further away, okay? But if we divide it into fifths, like a 5, do you remember what we would call this? It has a letter and a number. It's kind of like a really esoteric thing that I'm asking here, but
I don't remember.
You don't remember, right? But you remember sublamina A, sublamina B. But when we, and the reason why we do this is because if you look at it from the standpoint of the bipolar cell, for whatever reason, the bipolar cells are kind of like it's A or B. It's just sublamina A or B. But when you look at it from the point of view of the ganglion cells, for whatever reason, visual scientists broke it down even more granularly. Some ganglion cells end up in this part of sublamina A, and some end up in this part of sublamina B. And with regularity, it would be one or the other of sublamina A. And then here it would be like this part of sublamina B, and this part of sublamina B, and this part of it. And so it was in third. So we called this S1. Do you remember this? It was like S1, 2, 3, 4, 5. Yeah. And it's just to get you a sense that the IPL is so granular that's why when you look on the OCT, you can't really make out. It's not like everything is reflecting evenly. It's kind of like it's not like, you have this one big morass of cells that are just willy-nilly going. It's very specific. So every part of the IPL reflects almost evenly. So you don't get a perfect designation of that layer like you do for some of the other ones. You remember that?
Yeah.
Right. That's what I'm trying to say. When you look at the ganglion cell and the IPL, it's kind of looks like one big mush. It's because they all have perfect, kind of alignment, but enough that your light cannot tell you where there's a distinction because they're so granular. Anyway, so that's S1. And then which one is on and which one is off? Is this layer on or is this layer on? Which one is it?
The bottom one is on.
This one is on?
Sorry, I guess it's off.
Right. The bottom one is off. This one's on. So remember, further away, it's on. I'm just letting you know. It's just if it's something to remember. And that guy's off and that guy's on because he's terminating there. This is a ganglion cell, remember?
Yeah.
Okay.
Yeah.
And which type of cells are these ganglion cells? Good. Okay. Name the layer. Oh, now we're doing it in this other way. Name the layer and its contents. Okay. I'm pointing to that layer. What is that layer?
Layer.
Yeah. And what's its contents?
Ganglion cell nuclei.
You got it. Ganglion cell nuclei. What about that one?
The outer nuclear layer. The cell bodies of the photoreceptors.
You got it. Back to this one. What is that cell? FMB. It's inverted, right? Because a visual scientist drew this. In our parlance, these guys would be at the bottom of the image, but they're not because we didn't draw this.
So that's a midget bipolar.
Yep. And F means flat. So how many cones does it interact with?
Wait, no, one.
One, exactly. And is it on the surface or invaginating?
It's on the surface.
On the surface, right? Flat, midget, and it's an off bipolar cell. We know because it's in the off layer, which again is closer to where the bipolar cells are. You got it. One cone. Name the layer and its contents. There are 4 things in this. And what is the layer?
That's the internuclear layer.
Tell me the 4 things that are in it.
So cell bodies of the Mueller cells, amacrine cells, horizontal cells, and bipolar cells.
Perfect. Contralateral or ipsilateral? Which one is it?
That's contralateral.
Perfect. What? Now I'm really being like the boards, you know, I'm just hazing it up here. What are the cells called in the indicated layer? Oh my God. Tell me what are those cells? What is that layer of, what is this structure? Tell me.
I mean, is that the LGN?
It's the LGN. And then what is that layer if it's the colored layer there?
So that's going to be layer 1.
So what is the layer called? Layer 1, but it's right. It's the what are they?
Oh, it's the magnocellular.
Yeah, magnocellular layer. So it's magnocells or M cells, whatever. But I'm just trying to get you to remember that, like, it's not like the LGM's like a pass-through thing. Those are actually cells that sit there, and they're the magnocells or the M cells that then convey the information to the brain. Name the layer and its contents.
Nerve fiber layer. Axons of ganglion cells.
Perfect. All right, remember this. Okay, so I'm going to point. I'm going to tell you. Tell me, right? Tell me what type of ganglion cell is that?
That looks like it's bistratified.
Bistratified, which means it's both on and off, right? So what is the pathway again?
So that's coniocellular.
Which conveys what?
Information about blue light.
Right, information about blue light. Where is it project? Bistratified ganglion cell coniocellular pathway. Name the layer and its contents. That one right there.
Interplexiform.
Correct, interplexiform. So what is going on in the interplexiform? What 3 cells talk to each other?
So the bipolars and the amacrines and the ganglion.
You got it. Perfect. And it's the axons and the synapses between all of those. Exactly. Okay, back here, I'm going to do this guy. I'm going to tell you that's a small tree. You tell me if it's off or on. All right, off or on. Okay, and what pathway it would be, you know, going towards, and the information it conveys.
Okay, so that's probably a midget.
Yep, a midget. A midget what though? What midget what?
A midget ganglion
cell?
Yep,
midget ganglion cell. And then is it off or on?
Is it flipped here?
Yes, it's flipped. So which one would it be if it's flipped?
On.
It's on, exactly. So it's a midget on ganglion cell that would get information from some type of bipolar cell that was on. Hopefully it's a midget. Well, not hopefully, but maybe it's a midget, maybe it's not. But it is a bipolar cell that will synapse with it, that's on. And then what does it convey?
Red green light information.
Red green light, it's the parvocellular pathway, exactly, right? So midget on ganglion cell parvocellular pathway. Again, you're just like determining that from a random page in a book, which is just phenomenal. Okay, so this white layer, tell me what are the 3 cells that, what is it, and what are the 3 cells that interface in it?
So that's the outer plexiform layer, which has synaptic connections of bipolar cells, horizontal cells, and photoreceptors.
Perfect. You got it. Name the layer and its contents.
Vitreous.
Okay, and it's mainly water. Okay, that's fine. Okay, here we go. I'm going to point. Let me point to something here. Okay, I'm going to say central. If I say central, what is the color on the left that corresponds to the central visual field?
Blue.
Yep, it's the blue, right? It's the opposite, sort of like intuitively, everything's like kind of reversed, right? Makes sense? Cool. Rapid review. We're talking about horizontal cells. You're going to tell me where they're located, who do they talk to? So where are they located?
The cell bodies are located in internuclear layer.
Internuclear layer. So we said internuclear layer. And where do they synapse?
Synapse in the outer, the outer plexiform.
You got it. Outer plexiform layer. And with which cells do they talk?
Photoreceptors.
Photoreceptors, and then I'll just throw in there that, there are some bipolar cells innervation, but we'll talk about that later. Okay, primary visual cortex, we're almost there. I'm pointing to this layer, this top layer right here.
Let's see, okay.
What is that? Was that a statement?
I'm thinking.
Oh, I think you said M. I was like, okay.
I think that's a.
Is that alpha or beta? Right? Because we know that 4C. I'm pointing to like
4C. So that's going to be the the alpha.
You got it, right? Because remember how we're labeling, and when we count, we go 1, 2, 3, 4, 5. When we start, we go 8 with letters A, B, C, right? So this will be alpha, beta. So it makes sense if we're counting in that direction, we're going to do the alpha, beta in that direction. So alpha, 4C alpha, who terminates in 4C alpha?
So that's going to be magnocellular paracel type ganglion cells that convey information about motion.
Motion, perfect. Photoreceptors, where are their nuclei?
Outer nuclear layer.
Synapse where?
In the outer plexiform layer.
Yep. And then with who?
With bipolar cells.
Bipolar cells and horizontal cells, right? But you're exactly right. Okay. Name the layer. What is that one? Everyone loves this one now.
The myoid layer.
Yeah, myoid zone. And what is in that? Why does it look that way? What's in it?
The mitochondria of the cellular organelles of the photoreceptors.
Correct. Yeah. So like your endoplasmic reticulum, your ribosomes, all the things you remember in cell biology, right? That's that there. Just remember, myoid plus ellipsoid equals inner segment, but the outer segment is just under them and it has its own layer. Remember that?
Yes.
That's just the way it is. Okay. The inner segment has more of like a separation, almost like it went through a centrifuge. Okay. And you got the, the really, really collected ellipsoid organelles at the bottom, like a glass. And then on top, you've got this like, supernatant of like, endoplasmic reticulum and ribosomes, and Golgi bodies. But that's why you see the distinction. But that both of those together is the inner segment. And then right under it, you've got your outer segments, which don't have that level of like stratification. So they have just one layer. Makes sense?
Yeah.
Good. I'm pointing to this random line of Gennari. What does that correspond to which layer in V1? If you had to guess. What have we been talking? Every time I point to V1 and I show you that 1, 2, 3, 4, whatever, blah, blah, 4C, what is this?
It's probably a part. Wait.
I'll give you a hint.
Parvocellular?
No. So it's not parvo or magno. It's just where do both parvo and magno end up? In what layer? In the brain. So if I were to ask you parvocellular pathway in the brain in V1, where does it terminate? You remember? Because you said it to me before, it's 4C beta, remember?
Yeah.
Right? And then the magnocellular pathway terminates in 4C alpha. And that's 92%, like I used to like to use that line, 92% of all of your ganglion cells are terminating in 4C.
Right.
So layer 4 basically is that layer. You remember this?
Yeah.
Right? So that's why you can see it so distinctly from above, because it's not either parvo or magno. Parvo and magno, they do separate themselves when you really look up microscopically, but they're so precise as a unit into layer 4 that when you go from a bird's eye view and section the brain, you can see this. And why is this important? Because that's why we call V1 the occipital, the visual cortex. We call it the striate cortex because striate means stripe, and you can see 4C as a stripe. Make sense?
Yes.
Now you know why they call it that. Okay. Amacrine cells, where are their nuclei?
In the inner nuclear layer. In
the inner nuclear layer. Who do they synapse with? Give me their friends and where? Where do they synapse? Sorry.
They synapse with ganglion cells in the inner plexiform
layer. Inner plexiform layer, and also bipolar cells, I always say. So there's always like two, bipolar cells are involved in both, right? They're just like in there, but yes, exactly. Okay, name the layer and what's in it. I'm doing this layer that's sitting on top of the RP Brooks complex. What do we call that?
The interdigitation zone.
Interdigitation zone. And what's happening there? The apical processes of the RPE are kind of enveloping the photoreceptor outer segments, right? And then you get the shed discs, and that's how they recycle them. Perfect. All right, almost done. Here we go. I'm going to say that's a large tree. Okay, so like a larger, not a giant, but a bigger one.
Okay, so that is a parasol. or magnocellular ganglion cell.
Yep. And where was that, on or off?
It's off.
It's off. And then what is our, what does it convey again? Just remind me.
And information about motion.
Motion, right? Motion. You got it. Back here, what is that called?
The superior colliculus.
Correct. And then, so what is the class of cell that synapse is there? And then what does it deal with?
It's like the other ganglion cells that deal with saccades.
Saccades, exactly. Saccades. Perfect. Almost there. Ganglion cells, where are their nuclei?
The ganglion cell layer.
You got it. And then where do they synapse?
In the LGN.
In the LGN, that's correct. But what about in the retina?
Oh, in the inner plexiform.
You got it. Yeah, I mean, you were absolutely right. It's the inner plexiform layer. And then with which cells in the retina?
With the amacrine cells and the bipolar cells.
You got it. And then, of course, we know with the LGN cells, if they're or like the superior colliculus or the, pretectal olivary nucleus, all that other stuff, right? But exactly. So last, I think we're almost there. What is this one?
Okay, so that's layer 2 of the LGN, which is going to carry in for ipsilateral information from magnocellular or the paracel type ganglion cells.
Paracel type ganglion cells, you got it. And it's always motion. Okay, and then we're going to do, this one is giant. Who is the giant? If I said that they're the classic example of a giant ganglion cell, ginormous, it's just got one really big one. It's not a parasol. I'm giving you a hint. It's not a midget. It's not bistratified. It's that other one, but it's not an other because we don't know what an other looks like. So it's the fourth class that you know of.
It's the intrinsically photosensitive.
Photosensitive, right. And they deal with pupillary light response. They have melanopsin. Perfect. So IPRGC, suprachiasmatic nucleus, all the ambient kind of backup, the operating system sort of things that it deals with. Tell me that one.
So that's layer 4. It's going to be contralateral information from parvocellular or midget type ganglion cells carrying information about red-green.
Right. This is like so derivative now, right? You're like, come on, Dr. D, this is so easy. Bipolar cells, where are their nuclei?
Okay, so they're in the internuclear.
Internuclear layer. Where do they synapse? They have 2 synaptic layers in the retina.
In the inner plexiform and the outer plexiform.
Inner plexiform and the outer plexiform. In the outer plexiform layer, who are the 2 cells that they're friends with?
The horizontal and photoreceptors.
Correct. In the inner plexiform layer, who are the 2 cells they're friends with?
Amacrine and ganglion cells.
You got it. Perfect. Okay. Almost there. This one is a small tree.
So midget type cell, ganglion cell.
On or off?
That is off.
Off. And then what does it convey again?
Red-green light.
Red-green light. Perfect. Rapid review. Mueller cells, where are their nuclei?
In the internuclear layer.
Who do they synapse with?
Nobody.
Good. Which cells? Good. Yes, you're done. How was that?
Yay.
I mean, it's but
that was kind of fun, but also nerve-wracking.
Yeah, well, it wasn't you got everything right. I just want to point out, we've done a few, I don't know, 6 or 7 of these.
Yeah.
I would argue and you may agree with me or not, but it sounded like, if I show you something on an OCT now, I think you're going to be like, oh man, I just got that wrong. But I should know better. You get what I'm saying? It's no longer scary about the OCT. Now it's like, I don't know if it's sublamina A or sublamina B, or maybe it's a flat midget bipolar. You know what I mean? It's like such nuance, which makes this all the more useful, because the thing you're actually going to be, you should know for practice is the OCT. And that's the thing that I make, at least I like to think, pretty simple, relatively speaking, right? So very good job. This was not easy, but I always say whenever we do the quiz in the middle of it, I feel like it just makes you realize just how much you know.
This is great. Thanks, Dr. Dio.
Awesome. So then we'll go on to the next one. I think it's amacrine, if I'm remembering correctly. Okay.
Great. Okay.
All right. Okay. Bye.
Thank you. Bye.